Good afternoon and welcome to the fourth and final U.S. Food and Drug Administration's industry webinar to discuss the infant formula transition plan and exercise of enforcement discretion. Today's webinar will provide more in-depth information on the quality factor requirement of normal physical growth. I'm Lieutenant Commander Janisha Robbs with the FDA's Center for Food Safety and Applied Nutrition Communications and Public Engagement staff. In a moment, I will turn the call over to Dr. Patricia Hansen, Deputy Director of CFSAN's Office of Nutrition and Food Labeling, to provide opening remarks. Then you will hear from Dr. Andrea Lotze, Medical Director in CFSAN's Office of Nutrition and Food Labeling, Infant Formula and Medical Food staff to provide important information on FDA's requirements for recommendations regarding meeting the quality factor of normal physical growth. After the presentation, we will move to the question and answer portion of the call, where subject matter experts from CFSAN's Office of Nutrition and Food Labeling, Infant Formula, and Medical Food staff will answer questions that we have received through the registration process regarding the quality factor requirements and other details of the infant formula transition guidance. With that, I will now turn the call over to Dr. Patricia Hansen, Deputy Director of CFSAN's Office of Nutrition and Food Labeling.
Thank you, Janisha, and welcome everyone to the fourth and final webinar in this series. In this webinar, Dr. Andrea Lotze, neonatologist and medical director of our infant formula and medical food staff, will provide you with important information on FDA's requirements and recommendations regarding meeting the quality factor of normal physical growth. As I mentioned in the previous webinars, and everyone may be tired of hearing me say it at this point, but it is important, and so I will repeat it. The requirements for new infant formula submissions are the same, whether a manufacturer is eligible to pursue the path outlined in our infant formula transition plan guidance for its products, or a manufacturer is making a new infant formula submission for its products through our regular established process. This includes the quality factor requirements. Most of our recommendations regarding meeting the quality factor requirements will also be the same regardless of the pathway, although there are some process differences, primarily around timing. In the second webinar in this series, Dr. Carrie Assar provided a high-level overview of the entire landscape of FDA's new infant formula submission requirements and our recommendations, including the quality factors of sufficient biological quality protein and normal physical growth. In the second webinar, Dr. Krista Bew provided a deeper dive into FDA's requirements and recommendations regarding meeting the quality factor of biological protein, with a focus on the protein efficiency ratio, or PER, rat bioassay. In this webinar, Dr. Lotze will take a deeper dive into the quality factor of normal physical growth, with a focus on the growth monitoring study, or GMS. She will also touch upon the regulatory provisions that provide for exemptions from conducting a GMS, provided certain conditions are met. In addition, she will cover important touch points between the two quality factors, that is, sufficient biological quality protein and normal physical growth, including the relative timing of a PER and a GMS. We hope that all of you, regardless of the pathway you may be on and whatever your role might be in the process, will find this core information to be helpful. The information in this webinar may be especially useful to those involved in the design, actual conduct, statistical support, or oversight of a GMS study. And now, to turn things over to Dr. Lotze.
My name is Andrea Lotze, and today we are going to talk about how to meet the quality factor requirement for normal physical growth with a growth monitoring study, or GMS. I will begin by going over the definition of quality factors under 21 CFR 106.3. Going forward, italicized sentences are from the regulation. Under 106.3, quality factors means those factors necessary to demonstrate the safety of the infant formula and the bioavailability of its nutrients as prepared for market and when fed as the sole source of nutrition to ensure the healthy growth of infants. The quality factors for infant formula include the sufficient biological quality of protein and normal physical growth. It is important to note that the protein efficiency ratio PER rat bioassay, or rather an appropriate modification thereof, and the growth monitoring study, or GMS, are not the quality factors, but rather are the studies conducted to demonstrate that your new infant formula meets these quality factors. To provide a quick recap, Dr. Carrie Assar provided information about the new infant formula submission requirements and recommendations related to the quality factors in webinar 2 of this series, and Dr. Krista View provided details about the quality factor of sufficient biological quality of protein and the PER rat bioassay in webinar 3. Today, I'm going to talk about the other quality factor, normal physical growth, and the GMS. Before talking about the quality factor of normal physical growth and the GMS, I want to provide the reminder that it is a regulatory requirement in 10696F that the manufacturer conduct the PER rat bioassay on the whole infant formula and evaluate the results prior to initiation of the GMS. Prior to the conduct of the study required to meet the quality factors for infant formulas under 10696, we strongly recommend that the manufacturer ensures that all ingredients in the test infant formula meet the requirements of 10640A. Namely, the only substances that are considered safe and suitable for use in infant formula under the applicable food safety provisions of the Federal Food, Drug, and Cosmetic Act are those used in accordance with the agency's food additive regulations, are generally recognized as safe or GRAS for such use, or authorized by a prior sanction. An assurance of the basis on which each ingredient in the infant formula meets 10640A is a required component of a new infant formula submission in 106-120B6II. Manufacturers also need to ensure that all containers and closures, such as packaging, are compliant with 10640B, and we recommend that they do so prior to the initiation of a clinical study. The regulations in 10696C provide several exemptions from the requirement to conduct a GMS as specified in 10696B and may be requested for certain circumstances. I'm going to provide a brief summary of those exemptions, which include 10696C1 for changes to packaging of an existing infant formula, 10696C2I for an alternative method or study design, 10696C2II for changes to an existing formula that do not affect the ability of the formula to support normal physical growth, 10696C2III for market formulation in more than one form where the formula process using the method with the greatest potential for adversely affecting nutrient content and bioavailability meets quality factor requirements. Very importantly, exemption requests are considered by FDA on a case-by-case basis. Please refer back to the second webinar of this series for more detailed information about the exemptions. To begin, 10696A requires that an infant formula meets the quality factor of normal physical growth. To establish that an infant formula supports normal physical growth when fed as a sole source of nutrition, 10696B requires that a manufacturer conduct, in accordance with good clinical practice, an adequate and well-controlled GMS of the infant formula. The GMS for a new infant formula is a well-controlled study conducted in accordance with good clinical practice that uses anthropometry in healthy term infants at specific time points to measure physical growth together with formula intake in an age-appropriate amount. The minimum requirements for conducting a GMS are specified under 10696B as follows. Under 10696B1, the study is no less than 15 weeks in duration, enrolling infants no more than 2 weeks old at time of entry into the study. 10696B2 includes the collection and maintenance of data on formula intake and anthropometric measures of physical growth, including body weight, recumbent length, head circumference, average daily weight increment, and average daily recumbent length increment. 10696B3 includes the anthropometric measurements that must be taken at specific time intervals. The regulation requires that the measurements be taken at the beginning and end of the study, with at least 4 additional measurements made at intermediate time points, with 3 of the 6 total measurements made within the first 4 weeks of the study, and 3 measurements made at approximately 4-week intervals over the remaining 11 weeks of the study. Under 10696B4, the study must compare the anthropometric data for the test group to a concurrent control group or groups at each time point and compare the anthropometric data for each infant, namely body weight for age, body length for age, head circumference for age, and weight for length in the test group and the control group to the 2009 CDC growth charts. And finally, under 10696B5, the study must compare the data on formula intake of the test group with a concurrent control group or groups and a scientifically appropriate reference. We need to carefully consider good clinical practice and human subject protection. A GMS for a new infant formula is a well-controlled study conducted in accordance with good clinical practice that accurately measures at specific time points physical growth, formula tolerance, and formula intake of healthy term infants fed infant formula as the sole source of nutrition during the first 4 months of life. A GMS, therefore, needs to comply with the regulations for human subject protection for pediatric studies under 21 CFR Part 50, an institutional review board, namely IRB, review and approval under 21 CFR Part 56 to ensure that the infant study participants are not inappropriately exposed to risk. Throughout the study, the infant formula manufacturer, whether conducting the study or as a study sponsor, is responsible for its conduct, monitoring, and endpoints. In summary, quality control procedures that establish requirements to safeguard the infant's health and determine whether the new infant formula supports normal physical growth need to be included with appropriate documentation to the IRB for any adverse events and protocol violations. Next, we move on to study design as will be specified in your protocol. A GMS is a clinical trial that seeks to establish the safety of the test or new infant formula and its ability to promote growth compared to the control infant formula, which is an active comparator. For an infant formula to serve as a scientifically valid control, we strongly recommend using an infant formula that has been notified to FDA and lawfully marketed in the United States. Inclusion of the basic elements of study design, such as your hypothesis, participant selection, randomization strategy, data collection, including formula intake, monitoring, data analysis, adverse events, your statistical analysis plan, and how results and any adverse events will be presented and discussed are critically important. Attention to detail ensures that the GMS provides the quality and type of data to evaluate whether your infant formula supports normal physical growth when fed as the sole source of nutrition. There are several suggested study design checkpoints. Is the study design consistent with all requirements for a GMS under 10696B? Are inclusion and exclusion criteria appropriate? For example, have you included gestational age, birth weight, health status, age at entry, such as less than 14 days of age, birth status, such as singleton versus multiple, and informed consent? Is the collection of formula intake data part of the design of the study? Are the primary and secondary outcome measures appropriate? We suggest that the primary endpoint be the mean daily weight gain, namely weight gain velocity or grams per day. Secondary endpoints include, for example, body length for age, head circumference for age, and weight for length in the test and control groups. Z-scores, including weight for age, length for age, head circumference for age, and weight for length, are also very helpful, as well as formula intake. Are per protocol and intent to treat populations clearly defined? Note that the per protocol population includes the data generated by the subset of infants who complied with the protocol sufficiently to ensure that these data would be likely to exhibit the effects of treatment according to the underlying scientific model. Compliance covers such considerations as availability of measurements and absence of major protocol violations. The intent to treat group, which includes all subjects allocated to a treatment group, should be followed, with data assessed and analyzed irrespective of compliance with the planned course of treatment. Is the study sufficiently powered, e.g. 80% power, to detect a difference in the primary outcome, if a difference exists? Under 106.121A3II, a power calculation both prior to study initiation and at study completion is required. The regulations provide the minimum or basic elements of the GMS, while the study protocol provides all the operational details needed to successfully complete the study. The GMS for a new infant formula is a well-controlled study conducted in accordance with good clinical practice that accurately measures at specific time points physical growth, tolerance, and formula intake in healthy term infants fed infant formula as the sole source of nutrition during the first 4 months of life. Study protocol describes the who, what, where, when, and how of your trial. An essential first step is to provide a well-written study protocol that will meet IRB requirements. All analyses should be delineated in the protocol a priori with consideration of anthropometric measurements, namely body weight, recumbent length, and head circumference, formula intake, tolerance data, all adverse events, as well as any other endpoints, with the details contained in the statistical analysis plan, also known as the SAP. The protocol should pre-specify the coding dictionary that will be used, such as the Medical Dictionary for Regulatory Activities, known as MEDRA, with all terms consistently coded, including how the coded adverse events and any medication or dietary supplement use will be checked by study personnel for consistency and accuracy. Resources are available in FDA's guidance document detailing good clinical practice. The following are suggested examples of components to include in a GMS protocol. Please note that these are examples and not a comprehensive list. Did you provide the study sponsor, study investigators, and study sites? Are the background and rationale for the study included? Are the research objectives and hypotheses described? Are the design of the study, such as randomized, double-blind, and research methods included? For example, what are the primary and secondary endpoints of the study? How are participants selected? Namely, what are the inclusion and exclusion criteria? Is the randomization strategy described? Are study interventions and assessments described? Is your methodology for data collection and data monitoring described? Did the protocol specify how formula records are to be collected, reviewed, and corrected if necessary? Are definitions of protocol deviations included? Is your SAP included? We will talk more about statistical analysis later. I'm now going to make a few key comments about individual components of the regulations under 106.96b. The required time frame under 106.96b1 with enrollment of infants at no more than 2 weeks of age at time of entry captures the early critical infant growth spurt, while also maximizing time prior to when infants commonly will begin to consume complementary foods. Beginning the study with the infants' ages at 2 weeks of age or less and completing the study at approximately 17 weeks ensures that the formulas being consumed are the sole source of nutrition. The study groups should represent the population of infants intended to consume infant formulas. At least 2 study groups, namely control and test formula groups, composed of healthy term infants are required in a GMS. We recommend that participants of both sexes and in similar numbers be included in the study. Again, we also recommend that the study protocol clearly specifies the inclusion and exclusion eligibility criteria. It is important to note that it is not ethical to randomize an infant who is breastfeeding to be fed an infant formula. However, inclusion of a concurrent breastfeeding group of infants could be included as a reference group. In such circumstances, the growth of breastfed infants could also be compared to the groups of infants consuming the formulas as an additional comparator reference for normal physical growth. Suggested inclusion criteria for study entry might include, for example, healthy term infants, with term defined as greater than or equal to 37 weeks to less than or equal to 42 weeks of gestational age, birth weight between 2,500 to 4,500 grams and not older than 14 days at time of study entry, and a caregiver decision to formula feed the infant prior to study recruitment. Exclusion criteria might be defined, for example, to exclude congenital illness or gastrointestinal disease, or a strong family history of allergy to the protein source in the infant formula. Under 10696B2, the study includes collection and maintenance of data on formula intake and anthropometric measures of physical growth, including body weight, recumbent length, head circumference, average daily weight increment, and average daily recumbent length increment. Attention to detail and consistency in methods of data collection, whether paper or electronic, are critical to the success of a GMS. Most importantly, successful data collection is critically dependent upon concurrent data monitoring, to be covered later in this presentation. Typically, a minimum of 2 repeated measurements are obtained for each anthropometric measure of physical growth. An appropriate maximum allowable difference, or MAD, between repeated measurements should be defined in the protocol and readily available for personnel at each study site. Examples of MADs include 10 grams per weight, 0.7 centimeters per length, and 0.5 centimeters per head circumference. Slight differences between repeated measurements are acceptable if the difference is within the MAD. We recommend that an on-site decision tree be in place to manage errant measurements that fall outside the MAD. We also recommend that dedicated and identical equipment be used at each study site. Routine, frequent calibration of equipment, especially the scale, is critical. Under 10696B3, the study includes anthropometric measurements made at the beginning and end of the study, and at least 4 additional measurements made at intermediate time points, with 3 of the 6 total measurements made within the first 4 weeks of the study, and 3 measurements made at approximately 4-week intervals over the remaining 11 weeks of the study. This is a summary table that includes the visit and measurement number and when to obtain the measurements. We note that it is common for visits to vary from plus or minus 3 days around each study visit, and we recommend that you include this in your protocol. We also recommend that formula intake and tolerance data be captured and evaluated live at these measurement time points. Note that in our experience, we have found it optimal to also recommend collecting tolerance data while collecting formula intake information. Under 10696B4, the study compares the anthropometric data for the test group to a concurrent control group or groups at each time point and compares the anthropometric data for each infant in the test and control group to the 2009 CDC growth charts, which are incorporated by reference at 106.160. Note that plotted individual growth charts, including weight for age, length for age, head circumference for age, and weight for length, must be provided. Under 10696B4, we note that the study compares the data on formula intake of the test group with a concurrent control group or groups and a scientifically appropriate reference. The determination of what constitutes as a scientifically appropriate reference is a fact-specific inquiry, and typical formula intakes per the American Academy of Pediatrics are commonly used. Next, we will discuss study personnel. For selection of study personnel, we recommend that if selecting a clinical research team, also known as a CRT, you include a principal investigator, or PI, healthcare providers, dietitians, statisticians, and other research staff with expertise and experience in the conduct of clinical studies in neonates and young infants. If or when selecting a CRT, study oversight and appropriate data monitoring of growth at each visit and from visit to visit is ultimately the manufacturer's responsibility. We will talk more later about data monitoring and study oversight. For training of study personnel, it is essential to provide adequate training, monitoring, and evaluation of study personnel's ability to obtain and record accurate measurements and adhere to protocol requirements. We recommend live training programs prior to study initiation to provide a consistent approach adherent to the protocol for each of the anthropometric measurements to help assure measurement accuracy. We also recommend that each study personnel demonstrate and be evaluated on their ability to obtain accurate measurements. Continuous monitoring to confirm that study personnel are consistently following the correct procedures for collecting and recording data will help minimize variability in the study results. We recommend that retraining be provided as needed. We suggest training personnel to plot each infant's birth weight and subsequent anthropometric measurements on the growth chart during each study visit. This has distinct advantages, such as providing a visual aid for the plausibility of the measurements in real time, providing an opportunity for correction while the infant is still available for remeasurement if needed in real time, detection of notable growth faltering or excessive weight gain in real time. A clinician check can also be provided in real time if there is an adverse event report. We recommend that crossing of two major percentiles suggesting potential faltering or too rapid gain by any infant in either study group be clinically evaluated for growth issues during the study visit to determine if these issues could be associated with the formula or may have another explanation. We are now going to discuss data monitoring and oversight. We suggest that the data monitoring plan include frequent evaluation of the precision of each anthropometric measurement and consistent methods for managing measurements outside of the predefined MADs. A description of who can modify data values and under what conditions. For example, if 2 weights are required per protocol for each data point, addressing any greater than or equal to 10-gram differences between these weights with consistent resolution is critical to obtaining meaningful results. Suggested appropriate MADs between repeated measurements include 10 grams per weight, 0.7 centimeters per length, and 0.5 centimeters for head circumference. We also strongly recommend that the manufacturer and CRT perform data quality checks while the study is ongoing and make corrections with a clear audit trail, including what changes were made and by whom prior to database lock. The advantage of attentive monitoring is that it should be able to detect improbable values. If an electronic data capture system or EDC is used, it is optimal to have built-in edit checks to identify improbable data. Suggested anthropometric data collection checkpoints under 10696B 2, 3, and 4 include: does the protocol detail procedures to collect anthropometric measurements precisely and consistently? Does it specify the number of repeated measurements to collect and the MAD? Is there a plan or decision tree available for study personnel if repeated measurements are outside of the MAD? Have the measurement methods been tested to determine if study personnel perform and record the measurements accurately? Are individual data points plotted on the CDC 2009 growth chart during study visits? Under data monitoring, we also suggest anthropometric data clinical checkpoints. Upon review of each infant's growth chart, have study personnel screened for unusual growth patterns during the study visit, such as crossing of two major percentiles? Are possible explanations provided for infants with concerning growth patterns, such as an unusual increase, decrease, or plateau in growth parameters? Have concerning growth patterns been critically evaluated during the study visit to determine if, for example, potential growth faltering or too rapid weight gain could be associated with a formula related to formula intake or may have a clinical explanation? Are the clinical notes consistently present, and do they provide reasonably complete information regarding the infant's medical status? We would like to take this time to focus specifically on formula intake and tolerance, as our experience has shown us that collection of this information can be challenging. The relationship between formula intake and growth is basic to the evaluation of an infant formula's capacity to support normal physical growth. A 3-day record or diary of formula intake and tolerance collected prior to each study visit is often used for both test and control groups. Review of formula intake records by study personnel during the visit for completeness and credibility is highly recommended. Tolerance information, such as history of spit up, vomiting, stool frequency, consistency, color, number of times, and day of occurrence, is also optimally collected on the formula intake records. Caregivers should be provided with specific objective criteria to define tolerance outcomes. For example, there are tools available related to stool description, including color, consistency, number or times, and day of occurrence. Examples of tolerance events that may be causally linked to formula consumption include gastrointestinal upsets, such as vomiting, loose stools or constipation, respiratory manifestations, such as wheezing, and dermatologic reactions, such as rash or hives. We recommend that reports be evaluated by the PI, as the extent and degree of these tolerance issues may cause them to be considered as adverse events. Whether collected on paper or electronically, accuracy in collection of formula intake data is critical, and we suggest a few checkpoints prior to beginning your study. Have the formula intake records been tested before study initiation and during the study to see if they capture the information accurately? Has the method of collection been appropriately described in the protocol? Do the formula intake records include all necessary information, such as study ID, volume consumed, not the volume offered, day and approximate start time of each feed, and finally, is tolerance data included? Have you considered pre-specifying the conduct of sensitivity analyses, if necessary, in the event that there are observations that could potentially influence the study conclusions. We also suggest formula intake checkpoints during the GMS study. Except for the baseline visit, where data on formula intake collected for at least 3 full days prior to study visits, were data on non-study formula and/or food intakes also collected. Are formula intake volumes consistent with typical intake ranges for the ages of the infants at each study visit? Has formula preparation been reviewed and assessed? Are formula intake records complete? Additional formula intake data checkpoints include: are the minimum and maximum intakes plausible for the age of the infants at each study visit? Are any of the formula intake results skewed due to the outliers? Did you consider a sensitivity analysis with and without outliers to assess if potential errors in intake data affected study conclusions? We also suggest checking tolerance data for clinical correlates. Are there any correlations between unusual intakes and patterns of abnormal growth and/or adverse events? If there are clinical correlates, have they been carefully and adequately documented? As an example, an infant with a concurrent illness might consume less formula than usual. We are now going to discuss adverse event reporting. All adverse events or AEs must be recorded and submitted as required by 106.121A4 as follows. A report on attrition and on all occurrences of AE events during the study shall include identification of the infant by subject number and feeding group, and a complete description of the AE, including comparisons of the frequency and nature of occurrence in each feeding group, and information on the health of the infant during the course of the study, including the occurrence and duration of any illness. A clinical assessment by a healthcare provider of the infant's health during each suspected AE, and a list of all subjects who did not complete the study, including the subject number and the reason that each subject did not complete the study. Reporting of all AE occurrences during the study, the duration of the AE, regardless of severity, and relationship to the study product are required. Under criteria for AEs and clinical assessment, we recommend that criteria for AEs, including those for serious AEs, coding and recording, be applied consistently across all study sites by use of a coding dictionary such as MEDRA. Suggested criteria for a serious adverse event or SAE include that it is life-threatening and/or requires hospitalization. Clinical assessment by a healthcare provider during each suspected AE is essential to help determine if the event is related to the infant formula. We suggest AE reporting checkpoints under 106.121A4 as follows. Did you use an AE coding dictionary? Did you report the duration, namely beginning and end date of each AE, the severity, i.e. mild, moderate, or severe, seriousness, and the relationship of the AE to the test and control formulas? Did you compare the number or percentage of AEs in each particular coded category between the test and control groups and our individual AE tables included? Were you able to determine whether the test and control groups have similar types and numbers of AEs? Suggested adverse event reporting clinical checkpoints under 106.121A4 include: have the coded adverse events been checked by study personnel for consistency and accuracy? Are the clinical notes consistently present for each AE? And do they provide reasonable information regarding medical status that can impact formula consumption, collection, or interpretation of data? Do any AE results for the test group give you reason for concern regarding the test formula? Next, we will cover statistical analysis. For statistical analysis, the regulatory requirements for a GMS under 10696B4 and B5 include comparison of the anthropometric data for the test group to the control group at each time point, comparison of anthropometric data for each infant to the 2009 CDC growth charts, and comparison of formula intake of the test group with the control group and a scientifically appropriate reference. Statistical analysis under 106.121A3 must include statistical evaluation for all measurements, including group means, group standard deviations, and measures of statistical significance for all measurements for each feeding group at the beginning of the study and at every point where measurements were made throughout the study, as well as calculations of the statistical power of the study before study initiation and at study completion. Under 106.121A3II, as mentioned, calculations of the statistical power are required before study initiation and at completion. We recommend that the sample size be justified by, for example, providing information on the power calculation, attrition rate assumptions, and non-inferiority margin. Attrition is an important factor to consider when calculating the number of infants needed for an adequate sample size to achieve the appropriate statistical power. We note that attrition in infant studies may be higher than in other clinical studies and should be taken into consideration when calculating sample size. Sufficient statistical power is essential in order to detect a statistically significant difference in the primary outcome, namely growth, between study groups. Because an infant's weight gain is the most sensitive anthropometric measurement, weight gain is often considered the primary study outcome and could provide the basis for the sample size calculation for a GMS. The Statistical Analysis Plan, or SAP, is a technical document that describes in detail all aspects of the statistical analysis as outlined in the protocol. The SAP is often provided as a document separate from the study protocol, but we suggest that it be integrated and reviewed in conjunction with the protocol. The a priori clarity and details provided in the SAP, as with other aspects of the protocol, provide the guide for meaningful data analyses. For suggested details for inclusion in the SAP, we recommend that a statistically sound randomization process consistent with the protocol be provided. We suggest that at least 3 study sites be considered and that no single site provide more than approximately 30% of the total sample. We suggest that definitions of the per protocol and intent to treat groups be included. We also recommend that the statistical methods used for analysis be clearly specified. It is optimal for the SAP to include templates for the data listings, tables, and figures, as well as summary statistics on the data to be included in the final study report. Suggested statistical analysis checkpoints include: did an audit by the manufacturer and/or CRT take place to address implausible values or errors in the source data that could be corrected prior to locking data sets? Was a coding tool such as Medra used to code AEs? Did you include locked raw data sets with individual data for anthropometric measurements, formula intake, tolerance, and all AEs in your submission? Have you explained missing or implausible anthropometric or formula intake data? Did you include the required information under 106.121A4 for the AEs? Did you include information on duration and severity of the AE and relationship to study formula, if any? Are there a high number of missing data or protocol deviations? If so, are explanations provided? Considerations for the final report include an IRB-approved study protocol and SAP, a clinical study report summarizing all aspects of the study, including appropriate tables and discussion that includes the required regulatory information, as well as any relevant data resulting from a GMS that was well controlled in accordance with good clinical practice at appropriate time points, appropriate comparisons of outcome variables between the study and control groups, demonstration that the protocol was followed with discussion of protocol deviations, attrition and reason for discontinuation, robust discussion of the manufacturer's interpretation of the study findings and their conclusions, individual AE and SAE tables, plotted individual male and female CDC 2009 growth charts, locked raw data sets, and statistical analysis files. Recommended are examples of the case report forms, formula intake record form, any questionnaires, or study staff training materials used during the study. For closing comments, we suggest that you become thoroughly knowledgeable of regulations in 10696, including preamble to the infant formula interim final rule and infant formula final rule. While not required, we strongly recommend that manufacturers request FDA feedback on their protocol, SAP, and other study materials prior to study initiation and before IRB submission, so that any changes you may want to make after discussion with FDA will not need to be resubmitted to the IRB as an amendment. Throughout the study, the manufacturer, whether conducting the study or as a study sponsor, is responsible for its conduct, monitoring, and endpoints. We recommend that the manufacturer be actively engaged throughout study design, implementation, and completion to assure a meaningful outcome. We are also available for questions regarding any unanticipated issues that become apparent during the study. Of note, there's a great deal of information in the preambles about the quality factors as well as the GMS included among the reference resources. This concludes my presentation. Thank you for your time.
Thank you, Dr. Lotze. At this time, we will begin the question and answer portion of the webinar and answer several of the questions we received during the registration process regarding the quality factor requirements and other details of the infant formula transition guidance. As we cannot get to all the questions that were submitted, we will answer the most frequently asked questions along with any clarifying questions. Today we are joined by Dr. View, who will answer questions on the quality factor requirements. Our first question is, can you provide more clarity around the exemption from conducting a protein efficiency PER that is described in 21 CFR 106.96 G2? The regulation refers to changes to an existing formula that would not affect bioavailability of the protein, and that those kinds of changes could exempt a manufacturer from conducting a PER. What kinds of changes would qualify?
Yes. First, I want to be clear that in order to be considered for an exemption under 10696G2, the manufacturer needs to request that specific exemption and provide a detailed description of the change and an explanation of why the change made by the manufacturer to an existing infant formula does not affect the bioavailability of the protein. Second, I would like to clarify what is meant by an existing infant formula. FDA views an existing infant formula as an infant formula previously notified to FDA for the U.S. market by the same manufacturer requesting the exemption in the new infant formula submission. So now I will cover an example of a type of change that 10696G2 may address. So here an infant formula manufacturer proposed a major processing change to an existing infant formula. The firm requested an exemption under 10696G2, provided a detailed description of the processing change that included a comparison of the entire current and proposed manufacturing processes. The firm also provided a previous PER rat bioassay that was conducted on the existing infant formula, in addition to scientific support to show that the processing change would not impact the bioavailability of the protein or nutrients in the infant formula. And in this case, that scientific support included data and information related to the effects of the specific processing change to infant formula. Now, as a reminder, all exemption requests are reviewed on a case-by-case basis, considering the specific information that is submitted. And as mentioned in earlier webinars in this series, we will provide feedback to manufacturers who propose a specific exemption request from conducting a PER study.
Could you provide more clarity around the exemption from conducting a PER that is described in 21 CFR 106.96G3? The regulation refers to an alternative method to the PER that is based on sound scientific principles. What kinds of alternative methods would qualify? Does FDA have a list of alternative methods that could be used to qualify for this exemption?
So similar to the answer to the last question, in order to be considered for an exemption under 10696G3, the manufacturer needs to request that specific exemption, provide a detailed explanation of the alternative method, an explanation of why the method is based on sound scientific principles, as well as the data that demonstrate that the quality factor of sufficient biological quality of protein has been met. Now, importantly, the alternative method would need to demonstrate that the infant formula meets the quality factor of sufficient biological quality of protein, not just the protein source. FDA does not have a list of alternative methods that could be used for the specific exemption, but as mentioned previously, we will provide feedback to manufacturers who propose to request an exemption using a specific alternative method to a PER study.
What regulation states that the PER must be conducted prior to conducting the GMS?
21 CFR 10696F states that the PER rat bioassay shall be conducted on a formula and the results evaluated prior to the initiation of a growth monitoring study of the formula that is required under paragraph B.
Could you provide more clarity around the exemption from conducting a growth monitoring study, or GMS, that is described in 21 CFR 106.96C2II? The regulation refers to changes to an existing infant formula that does not affect the ability of the formula to support normal physical growth, and that those kinds of changes could exempt a manufacturer from conducting a GMS. What kinds of changes could qualify?
So as mentioned in response to the question regarding a similar regulatory exemption from the requirement to conduct a PER study, in order to be considered for an exemption under 10696C2II, the manufacturer needs to request that specific exemption and provide a detailed description of the change and an explanation of why the change made by the manufacturer to an existing infant formula does not affect the ability of the formula to support normal physical growth. And I would like to reemphasize what we mean by an existing infant formula. FDA views an existing infant formula as an infant formula that was previously notified to FDA for the U.S. market by the same manufacturer requesting an exemption in the new infant formula submission. So next I will cover an example of a type of change that 10696C2II may address. An infant formula manufacturer proposed a major formulation change to change the source of a nutrient in an existing infant formula. The firm requested an exemption under 10696C2II, provided a detailed description of the formulation change, and a detailed explanation for how the change in the source of the nutrient would not affect the ability of the proposed formula to support normal physical growth. The firm referenced their previously submitted growth monitoring study for the existing formula and provided a discussion of data and information from published clinical studies that were conducted using infant formula that contained the newly sourced ingredient. Now this is just an example, and all exemption requests are reviewed on a case-by-case basis. And as mentioned previously, we will provide feedback to manufacturers who propose a specific exemption from conducting a growth monitoring study.
Could you provide more clarity around the exemption from conducting a GMS that is described in 21 CFR 106.96C2I? The regulation refers to an alternative method to the GMS that is based on sound scientific principles to show that the infant formula supports normal physical growth in infants when the formula is fed as a sole source of nutrition. What kinds of alternative methods would qualify? Does FDA have a list of alternative methods that could be used to qualify for this exemption?
So in order to be considered for an exemption under 10696C2I, the manufacturer once again needs to request that specific exemption and provide a detailed description of the alternative method or study design, an explanation of how it is based on sound scientific principles, as well as the data that demonstrates that the quality factor of normal physical growth is met. With respect to the exemption 10696C2I specifically, the alternative method or study design needs to be based on sound scientific principles. These would include things such as an appropriate and scientifically valid control formula and sufficient study enrollment to ensure adequate statistical power. Now, importantly, the alternative method would need to demonstrate that the infant formula meets the quality factor of normal physical growth when fed as sole source nutrition, not just as the primary source or one of several sources of nutrition. Furthermore, a manufacturer that intends to conduct a growth monitoring study that does not fully comply with the specific requirements in 10696B must request an exemption under 10696C2I. FDA does not have a list of alternative methods or study designs that could be used for this specific exemption, but again, we will provide feedback to manufacturers who propose to request an exemption using a specific alternative method to the growth monitoring study as described in our regulations.
Great. Thank you so much, Dr. View. Now we are joined by Dr. Hansen to answer several of the questions we received during the webinar process regarding other details of the infant formula transition guidance. First question, what happens after November 14th? Can manufacturers still make a request for enforcement discretion? Is FDA going to be issuing any additional letters of enforcement discretion? Will FDA respond to firms that made a request for enforcement discretion but will not be receiving a letter of enforcement discretion?
Well, now that November the 14th, 2022 has come and gone, FDA will no longer be accepting and reviewing requests for enforcement discretion under the May 2022 Enforcement Discretion Policy Guidance. FDA is in the process of responding to those firms who made a request for enforcement discretion, pursuant to the terms outlined in that guidance and made it on or before November the 14th, and to which we have not issued a letter of enforcement discretion. I will say we are working to issue those responses as soon as possible.
We currently have an equivalent to the FDA approval in Australia. If we do not receive a letter from the enforcement discretion by November 14, 2022, are we still a part of the program? Can we start to market and sell our product?
Now, I want to be very, very clear on this point. If you do not receive a letter of enforcement discretion from FDA in response to a request and supporting information you provided in response to FDA's May 2022 guidance, then you are not a covered infant formula manufacturer under the terms of the September 2022 Infant Formula Transition Plan guidance, and you are not eligible to pursue the pathway or process outlined in that guidance. You are, however, welcome to pursue entry into the U.S. market via our normal established regulatory process for new infant formula submissions. So please see webinar 2 in this series, recorded and posted on FDA's website, for an overview of FDA's requirements and recommendations for new infant formula submissions. The last couple of slides in that presentation will also provide you with a list of resources and links that will provide more detailed information.
For those companies that already received enforcement discretion, do they need to ship their first batch of infant formulas to the U.S. by January 6th and follow the steps stated in the transition plan to undergo formula registration to remain in the U.S. market?
For those firms that receive or have received a letter of enforcement discretion from FDA under the terms of the May 2022 guidance, the period for which FDA intends to exercise continued enforcement discretion for the products specified in that letter has been extended to January the 6th of 2023. Please see page 7 of our September 2022 Infant Formula Transition Plan guidance for that. FDA's decision whether to continue to exercise enforcement discretion after January 6th will be made on a case-by-case basis for covered infant formula products that are specifically identified in a letter of intent submitted by a covered infant formula manufacturer to FDA and that FDA has acknowledged, consistent with the policies described in FDA's September 2022 guidance. Please also see phase 1 as described in that guidance. And then for FDA to consider the further exercise of enforcement discretion for covered infant formula manufacturers and their covered products. As a first step, those manufacturers need to submit that letter of intent to FDA by December the 5th of 2022. And in that letter, they need to request continued enforcement discretion for one or more of their covered infant formula products and state their intent to pursue completion of all regulatory requirements for those specific infant formula products.
Do you have plans to add to your website those manufacturers who have submitted letters of intent?
That's a good question. And similar to the way in which we have provided information about infant formula products for which infant formula manufacturers received letters of enforcement discretion under the terms of the May 2022 guidance, FDA also plans to provide information on its website about infant formula products for which FDA intends to continue to exercise enforcement discretion under the terms of the September 2022 Infant Formula Transition Plan guidance. So yes, we will post information about those who will continue to receive enforcement discretion up on our website. Transparency is very important to FDA.
Can interim changes be made, for example, labeling, packaging, formulation, potentially driven by supply chain disruptions, during a transition period for products currently imported under enforcement discretion?
We encourage manufacturers who are experiencing supply chain disruptions that may prompt consideration of these kinds of changes to contact us, please, and we will work with them directly. Some changes may be sufficiently minor that they can be handled as an amendment to the original enforcement discretion request or letter. and other changes may be significant enough that they cannot be handled in this manner and may be out of the scope of the September 2022 Infant Formula Transition Plan guidance. In the event that they are out of scope, those changes should be handled via the normal, regular established process for new infant formula submissions.
Great. Thank you so much, Dr. Hansen. Thank you, Dr. Lotzi and Dr. View. This concludes today's webinar. A recording will be available on the FDA's meeting webpage shortly. Thank you and goodbye.